Hi, everyone. Thank you for joining the webinar. We're just going to wait a few minutes for people to connect. Hello, good afternoon, and welcome to our webinar. My name is Yael. I am part of the marketing team here at Energene, and I'll be your host today. In today's webinar, we'll be discussing the challenges of raw materials in the alternative protein industry and how these challenges can be overcome through specific crop breeding to impact and improve their entire value chain. We will save time for questions at the end of the webinar, so we encourage you to type in your questions during the presentation using the Questions tab on the left of your screen. This webinar is being recorded and will be available on our website in case you would want to listen to it again. It's my pleasure to present today's speakers. Our first speaker is Dr. Tom Ben-Aryeh, a senior scientist at the Good Food Institute Israel. Dr. Aryeh provides scientific counseling to the alternative protein ecosystem and provides courses on alternative proteins at the Hebrew, Tel Aviv, and Ben Gurion universities. The Good Food Institute Israel is an international science-based nonprofit organization focused on building a sustainable, healthy, and just food system. I would also like to welcome Dr. Shannon Hood-Neifer, Vice President of Innovation and Technology at the Food Center in Saskatchewan. Shannon has worked in the food industry for over 20 years, gaining experience in developing new food products, assisting entrepreneurs to start food companies, and implementing innovative solutions in food companies. At the Food Center, her primary role is to strategize technologies to be adopted and to help food companies work with non-traditional ingredients and innovative solutions. Lastly, we have our very own Dr. Uri Weishaus, who has been with Energene since 2011, applying genomic tools for marker development and breeding projects for various crops. Uri holds a PhD in plant and microbial genetics from the Hebrew University, and his academic background is in genetics of plant secondary metabolism pathways. So we will begin with Tom, and the stage is yours.
my screen. Oh, excellent. So thank you again for inviting me. Today I will talk about alternative protein with a little bit of focus on crop optimization and plant-based products. So when we talk about alternative proteins, we usually talk about three main technologies: cultivated meat, plant-based meat, and fermentation. However, for today's talk, we will focus on the plant-based meat side, on the plant-based proteins. So why do we need alternative proteins? Intensive factory farming and poor animal welfare are a cause for foodborne illnesses, such as the swine and avian influenza. They also contribute to the spreading of E. coli, Salmonella, Campylobacter, which can be found in meat. In addition, overuse of antibiotics are used in the animal industry are basically the overuse of antibiotics that are given to the animals in the industry can also generate the antibiotic resistance to those antibiotics in a bacteria, which is something that could be very harmful for human health. It is estimated that the OECD countries will spend over $2.9 trillion by 2050 on antibiotic-resistant bacteria, and estimates show that antibiotic-resistant bacteria will kill more people than cancer by 2050. So this is a threat that is going to happen to humanity, and we need to find ways to reduce the use of antibiotics. For example, antibiotic resistant to colistin originated in 2015 in pig farms in China. Colistin, the last resort antibiotics, and afterwards they found it also in chicken and pig farms in Vietnam and South America. And just to show how it also gets to human. A few years ago, there was the first case in which a woman was hospitalized in Nevada, and her physicians found in her body bacteria which were resistant to all known antibiotics. There was no way to treat her, and she passed away. And our thought is that if we can reduce the use of antibiotics, we will see less, and we hope that those cases won't increase over time. So basically, the goal to use less antibiotics, which can be that we can get to that by using plants over animals. In addition to that, the environmental impact, I think you all heard about this. I will not go over everything, but I will just mention that animal agriculture pollutes more in terms of greenhouse gas emissions, more than all of the transportation combined. And that in terms of resources, in order to generate 1 kilogram of beef, you need 15,000 liters of water. Just to make sense, just to understand what this means, our domestic use of water is about 5% of the water use of our population, whereas animal agriculture uses 55% of the water. In addition to that, meat production demands 40% of the arable land. And by 2050, the demand for meat will double. So basically, we basically trying to say that the current way that we are producing meat is not sustainable, and we need to find a more efficient way to produce it. Lastly, another motivation for alternative proteins is that what happens to the animals in the industry. This industry slaughters over 100 billion animals per year in ways that most people don't even want to hear about, which is another social reason why this field has interest. So when we talk about plant-based meat, our main goal is to make plant proteins to act as animal-based proteins, where the plant proteins are usually globular proteins, and whereas the proteins that we aim for are more fibrillar ones. And we have different techniques of how to do that. this is where most of the focus is. In this lecture, we will talk about different opportunities that we have in the plant-based meat industries. We can divide it into several categories: the source of the plant protein, perhaps optimizing those plants, which we will discuss briefly. In addition, there are ways of improving the raw materials by the way that we process them, and also generating final products. In this lecture, we will focus mainly on the source and the optimization of the proteins. So what proteins used in this industry? Mainly soy, as you already know. However, there is a increasing use of pea protein, and also wheat protein is also out there, and many other ones that have some that you don't use them in bulk. However, they have interesting properties. We at GFI generated this new research, the plant protein primer, that gives you a lot of data on many types of proteins, because it's really complicated for a new startup to choose the right protein source. So we made a list of 19 proteins that are currently in use, and gave quantitative measurements of different parameters important for those proteins, such as health, measured by protein concentration and TDCAS, or sorry, not health, but nutritional value, allergenicity risk, a commercial stage of using that protein for meat alternatives, the flavor of this protein, which we hopefully have no flavor, the functionality, what kind of products, what kind of properties do those proteins can have, whether it's gelling and emulsification and so on, the cost of the protein, and the global volume of production. So this is a nice resource. You are welcome to ask. We will be happy to share it. We also generated for each protein one slide that explains much more in depth about that protein. As we see here with pea, it's the fastest growing plant protein. The sources, the nutritional aspect. For example, the nutritional value is rather okay if we look at pea, and however, it still has some anti-nutritional factors such as lectin and trypsin inhibitors, lectin, trypsin inhibitor, proteolytic acid, and so on. You can see that the protein is rather nice, 28% protein. However, it has lots of fiber and carbohydrates. So, and we need to understand how to use them. Whereas in soy, you have lots of oil, which is easily used, and so on and so forth. You can really see the comparison between soy and pea, for example, and understand why some companies decide to move for soy. Soy has many benefits. Still has one of the 8th most allergenic plants out there, and it has some distinct flavor that is not the best. If we talk about crop optimization, which at least in our mind, or at least in my mind, one of the major ways in order to advance this industry, basically most of the projects out there to develop new products are going towards the raw material and optimization, developing new ways, new techniques in order to make the products more similar to meat. However, the problem is that each of these processes make the products itself much more expensive. Whereas if you take the plants and you make them more similar to meat, you basically improve the quality without making any effort for each individual product. And we think that this could really revolutionize the field of plant-based meat alternatives, or eggs, or dairy alternatives. Some directions that could be rather simple to perform: increasing the protein concentration, increasing the iron concentration, playing with the amino acid composition, perhaps introducing with genetic modification some meat flavors. Those are all very simple directions. However, the interesting directions that we are looking towards is improving the plant as a raw material for the processing. For example, increasing the water holding capacity of the plant, increasing the oil holding capacity, and lowering the variability of the plants. So these are just a few directions that we think that could be valuable for this specific industry. I will just mention the Good Food Institute, as I mentioned, is the Good Food Institute is a non-profit organization, science-based, with over 100 staff, working in three directions: science and technology, corporate engagement. In the science and technology, we work with universities, academia, and so on, providing courses, giving grants for open access research. We also have the corporate engagement, work with the top companies and working with startups in order to develop this field. Policy working with governments, generally regarding regulation and funding for research. We have branches all over the world, in the US, Brazil, India, Israel, Europe, and Asia Pacific. And if you are interested in the field of alternative proteins, we will be happy to help with resources and so on. So thank you very much for listening. I think that the next lectures will be really exciting in terms of crop optimization and generating plant-based alternatives. And I'll hand it over to Shannon. So thank you for listening and have a great evening or day.
Excellent. Thank you very much, Tom. Okay. Siri. Okay. Well, I'd like to thank Energene for inviting me to speak today. I'm Shannon Hood-Neifer, VP of Innovation here at the Saskatchewan Food Centre, here in Saskatoon, Saskatchewan, Canada, where we're a not-for-profit organization that helps food companies develop new products and processes for food or ingredients. We also have food safety training, as well as ingredient innovation and some manufacturing capacity here, where we have access to over 280 pieces of food processing equipment to do a multitude of things, including plant-based proteins and plant-based product development. And so we have, in this line, we have extrusion, we have a variety of drying techniques, an ingredient extraction and alteration of functionality, and some fermentation capacity. But we're here to talk about plant-based foods, which, you know, I've been at the Food Centre for 11 years now, and we've been making meat alternatives for all that and then some. And so we've put in a considerable amount of energy and expertise into both the extrusion side, but also the post-extrusion side. So how do you turn plants into meat analogs? And so we're taking plant-based proteins and plus some other plant materials. So depending, in this particular photo, you're looking at what is a high moisture extruded product. And so typical ingredients in this would be a plant-based protein, a starch, a fiber, and some flavors, some oil, depending on the texture that you're looking for. And so to be able to take, as Tom alluded to, you're taking these globular proteins, and then you need to transform them into something that's fibrated and meaty-like. It does take a little bit of ingenuity, but totally not unachievable. And so here at the Food Centre, we use co-rotating twin screw extrusion. We do have a Klextrol setup. That picture is of me almost 10 years ago, and so I look much older now. But we are running. So where you can see my hand, where the feed throat is, and then the dry material comes into the extruder, and the screws are turning very fast, and then we are heating and we're cooking the material. And then the photo on your right, or the depiction on your right, you have these globular proteins that are now going to unravel due to shear and heat. They're going to align with the flow of the extruder, which is a laminar flow. And so then these proteins will align and then they will bond together as they cool. And this allows us to do a number of great things. And so types of ingredients that we would put into here and potential protein sources, as Tom had alluded to and Ori will talk about in a bit, we're using wheat, soy, corn. We've tested canola, oat, pea, chickpea, flax, mustard, lupin, pumpkin seed protein from pumpkin seeds, hemp, potato, rice, sachinchi, algae, and then I'm adding here crickets and mealworms. They don't fit into plant-based, but they are an alternative protein source that is being investigated at this stage. And there's pros and cons to each one of these. And so one of the things that we need to consider is, you know, the functionality, the flavor differences that you're looking for as you move through your product development and your manufacturing stages. So some of the challenges, as you could probably see from the list and from the earlier pictures, one of the challenges is color of the protein. And some of this is based on seed. Some of this is based on how that product or that protein has been extracted and dried. It may change some color. And so typically, so say if you're using canola protein, which tends to be a bit darker, you know, meat products typically aren't that dark. And so you might need some color, might need to adjust color. Texture, you're looking for a variety of different textures. And this is the fun part about working in plant-based meat analogs, where there's just such variety and such differentiation among the textures that we're looking for, whether you're mimicking beef or chicken or pork or seafood, or the wide varieties of products in those categories. So sometimes you want it chewy, sometimes you want it, you know, bite, sometimes you want it flakier, but you don't want it overly chewy. Sometimes, depending on what we're trying to do, we might get a product that's gummy. You're looking for different degrees of fibration, and we can adjust these with formulation and extrusion operating conditions. But as we work through some of these new feedstocks, you know, I think some of these solutions can be through breeding all the way through processing, and then so we can do innovation through the full life cycle of a crop. Some cost, so some of these new emerging proteins, as Tom had alluded to, can be costly as they scale. Then also as they scale, availability of the emerging proteins, and so sometimes you find the perfect protein and it might be back ordered, or the company hasn't quite come online yet, and then so then you have a bit of a conundrum, a chicken egg thing, where your commercialization capacity is limited, is hinging on somebody else's. Consistency of your raw materials, so difference, we see a fair bit of lot to lot variation, which tends to lend to thermal history. So different changes in your drying of your proteins can affect this type of extrusion. But the biggest one, I've saved the last two big ones for last, functionality. In this particular scenario, we have proteins that need to function in a specific way. They need to unravel, they need to align, and they need to be able to align with the flow of the extruder, and they need to be able to bind together. And some proteins lend themselves to be able to bind in certain matrices better than others. And so we try to select functionalities according to that. And this is something that we can work on through breeding. And then flavor. We're looking for proteins that are blandish. And I say blandish because sometimes that background flavor can be beneficial. But these proteins tend to taste like plants, which if you're trying to mimic meat, then tasting like plants isn't what you're quite looking for, although there's a whole entire category of products that taste like plants, and that's fantastic. And sometimes the proteins are bitter, and so sometimes you were looking at, you know, removing the bitter flavor in advance, or using maskers through the extrusion process, or using other combinations of ingredients to control the bitterness. And so this is the protein's contribution to flavor in these types of applications is something that needs to be overcome. But with that being said, I'm going to end on a few slides of some photos of some products that have been made. So we have pulled pork, barbecue pork ribs, and ginger beef, and all of these are full vegan. And most of the formulations in these pictures, the two pork ones will be probably a soy and a gluten blend with a tiny bit of gluten in it, and the ginger beef is pea and soy blends. And then this is a vegan beef empanada. And then sausage. And so we have some pepperoni sticks here. And this particular technology is more challenging because you're trying to pull together already denatured proteins into a matrix that will look and taste and feel like sausage. So this is an older generation of our vegan pepperoni sticks. And so we have a newer generation that looks slightly different, but this is the nicest picture. And so that's all I had, and I will turn it over to Uri. Thank you very much.
Hi everyone, this is Uri. Thank you, Tom and Shannon, for the previous talks. I'm going to try to connect all of these topics together and discuss how, and hopefully convince you that we can solve a lot of the challenges in this emerging, interesting new field by treating everything right from the start, as our logo says. And we get distracted here, and we aim to use all of that in the breeding process. All right, so I'm going to discuss a few topics here, mainly about how breeding interacts with the entire plant-based protein value chain. I'm going to discuss the value chain, the components of it, and why breeding can help and improve all the steps in this process, and then show you how the breeding can provide solutions that treat a lot of the aspects of the raw material and the processing steps, and then a quick introduction about how Energene delivers quick solutions through breeding, conventional breeding. And might I just add that the previous discussions have all mentioned alternative meat products. An emerging new topic that is also very suitable for this discussion is alternative seafood replacement or seafood protein as alternatives, plant-based sourced to produce seafood-like alternative. Same principles apply in this case as well. So quick word about Energene. We're a genomics company that's been around since 2010 with multiple products and services for a vast number of clients, mostly in the plant-based space, seed and plant-related products, but not only seeds, also end products that are processed from plant-sourced material. We've handled over about 80 species so far, genomic-wise, and published over 25 publications in the best peer-reviewed publications. So the technology is very much validated by both industry and the academic world. So as I said, the plant-based meat value chain has several steps, and these steps are first contain the IP that you bring with you, start with, or purchase. And then there's the input materials and steps involving actual creation of the protein, and then processing and formulation, and finally distribution. So in this webinar, I'm going to I claim that we could interfere and improve all steps related to this value chain. Not all of them in each point, but at least one aspect of each of these steps we could improve through breeding. In the terms of IP, functionalization and formulation, a lot of the know-how related to producing alternative proteins starts with protein. So if we, with plant-based proteins, so if we improve the source, the raw material, we have new IP. It's very easy to protect versus other types of technology. So if you have a seed, you register it through plant protection programs, and the IP is very well protected, but also you have control of the source of the seeds. We mostly use hybrid seeds, so those are also IP protected and also a trade secret. You keep the parental lines, and when you have such leading plant material, part of your IP is unique, meaning if you have access to a particular pea or lentil variety, only you could use that process. So that's one example of how IP is influenced through creating better seed varieties. And then the input materials, every aspect of that can be controlled and improved by breeding, whereas the protein source, it's the multitude of traits that were mentioned by the previous speakers, texture, structure, and so on. The content and value of the protein content versus other constituents. A lot of people talk about protein content, but fail to mention the other aspects of the crop. If you're using canola, canola is a very important oil crop, and so goes on for other legumes that are significant sources of starch. So you need to balance between the protein content and alternative uses of the same crop. And then there are other aspects of the input materials, such as binding, flavors, colors that were mentioned by Shannon earlier, and many others that could all be addressed through breeding. And then also creating the protein. I'd mentioned that the production facilities are usually tuned for some sort of operation. When you change a major feature of the crop, for example, the starch content, you change the weight of the actual program, you change the machinery required for it. So you need to interact and work with the producers. And you're actually changing the entire value chain at this point. For example, from pea, sometimes as much as 60% of the pea is starch. So if you're changing the content of pea to improve texture, for example, then you are changing also the value and the processing technology required for pea. So that's one consideration that you need to consider, and we claim that you could actually address that in the breeding process. And then the processing were also addressed earlier. Texturizing and flavoring. Packaging is not very relevant here, but the part of the value chain that touches the processing is super important. Flavor was mentioned a lot, but also texture is now becoming one of the key traits to address in the breeding. And eventually sales is not something directly impacted from breeding right from the start, but the entire value chain is influenced. And if you're changing the entire value chain of plant-based proteins, you obviously affect sales as well. So the example that I want to show in this case, mostly from work that had been done in the last, I would say, decade in Australia, is taking the lupin plant, which in theory is the ideal superfood crop. As you can see on the left, most of you know lupin as an ornamental crop. It grows wild, it grows domesticated, very pretty plant with a very, very significant percentage of protein, very nutritious, both for feed and for human consumption. There's a subtype called narrowleaf lupin, and that subtype can be converted from a wild species, or was converted from almost a wild species into a domesticated crop, and a very successful one. I'll touch the problems and values of this crop in a bit. So the sweet lupin, as I said, it's the ideal legume. It is more rich in protein than quinoa, more dietary fiber than oats, and has more vitamins than other, and bananas and iron and so on. So in theory, it's the super crop. is very, very well accepted as a crop, but it has a few downsides. For examples, in some cases, it's an allergen, much similar to peanut allergens. And there's also some alkaloids in there that need to be bred out, meaning through non-GM technology, if you have the right genomic tools, you could breed against and get rid of the alkaloids that are a problem and prevent this highly nutritious 40% protein legume. So this had been done to some extent in Australia, but it took a long time. And we offer a process that could achieve this much faster. So one of the bottlenecks of breeding lupin, for example, had been genomic tools. And in recent years, there are two bottlenecks or three bottlenecks. One, the genomic data. So that had become quite cheaper to make, but the analysis became more expensive as more and more data gets accumulated. These nice images actually are of the, they come from the publication earlier this year of the lupin genome. So not only did they make one lupin genome, they actually sequenced 15 accessions or 3 groups of white lupins. So we have capabilities now to create a lot of data for a lot of crops. The computational challenge, actually handling that much data, becomes very challenging. And also the analysis. You need experts to analyze and get high value from this large masses of data. The end goal of the entire process, and this is what Energene specializes with, is collaborating with a lot of people involved in the breeding program and in this whole value chain that I showed to create this ideal crop in one go. Generally, this is referred to as trait stacking. With the genomic tools that Energene has developed, we can, and when we do in collaboration with others, genomic projects to isolate, locate specific traits, all of them in a lot of genetic, several genetic backgrounds and genomic tools. And we can map and create genetic markers that enable integration of all these traits in, I wouldn't say in one go, but in a process that takes fewer years than the decade I showed earlier for lupin. And we could influence, together with food technology experts like Shannon and the GFI and other organizations, we could integrate into the breeding process the entire, I would say, value chain interpretation integrated into the breeding process so that we could analyze and incorporate genetic markers and create the ideal processing lupin bean. So Energene can do that, not specifically for lupin. We haven't done this in lupin, but we could do this for a lot of other crops. Legumes are usually the most kind of the immediate suspects because there's a lot of genetic diversity, they're well accepted and they're well used, and the industry is tuned to process legumes. And currently most of the plant-based proteins are to some extent based either on pea or soy or combinations shown before of legumes and others. So we can examine a broad range of cultivars, detect the genetic diversity, process that data, transfer that into desirable traits, and create new cultivars that will generate new IP with better nutritional values and a lot of the desirable traits for this value chain. One of the key components that I'm offering here to do, and this is really a call for collaboration, is integrated breeding, the application of formulation technologies in the breeding process. We take in small batches with the help of the experts in the field. This long list of traits are all important and crucial to plant-based alternatives. So what we can do, and what we're doing in some cases, is take the list of traits, not care about protein content only, but take all this list of traits, prioritize them, and integrate them, or at least the major influencers of the texture traits, integrate those into the breeding process, so that in one process we could breed for both protein content and yield, which are always desirable, but also combine into the process the major influencers of the texture that influences in the end the value of the product as seeds and as processed seeds. We have done, for example, such a database for soy. Energene has already set up a vast database, probably the largest at this resolution, of multiple genomic lines or multiple genomes of soy. So we have this database that allows us to take any type of soy not in the database and currently in the database, examine the genetic content, associate genotype and phenotype, and use other genomic tools to eventually create vast array of flours or processing meal to be used in the development process of alternative meat products. Another example of something that we are involved with is an end product that is not necessarily legumes. As you can see here, tires for rubber substitutes. These tires are not made from synthetic rubber. Some of them include natural rubber that does not come from the rubber tree. Alternative sources exist. This is a plant called guayule. So we are taking guayule plants and converting those in relatively short time compared to a breeding program into a crop that could be used to harvest rubber substitutes in the entire process to create high durable and valuable products. So to sum up, we could do this entire process for a lot of crops. The immediate suspects are the ones currently used for food and feed, peanuts, legumes, and cereals, but also we could integrate this process with a lot of other products that can be used either as a source of alternative protein or as additional for flavor or color and so on. And we can integrate the breeding cycle with the processing experts and convert all of that into one successful short duration project to create new meat alternative products. With that, thank you all for listening, and we'll be taking some questions and hoping for you to reach out and offer us collaborations in specific projects.
Thank you, Uri, very much. So now we have time for a few questions. Thank you to everyone in our audience who sent us questions. So the first one is for Shannon. Are there any negative aspects to the plant processing in terms of nutrition that we need to make our customers aware of?
That's a really good question. Well, I guess some of the things that need to be highlighted or addressed would be that as you're extracting protein from a seed, that you are taking away something. Typically, depending on the type of fractionation or isolation that you're doing, you're going to lose some of your micronutrients, and so you need to be aware of that. And in some cases, depending on where you are in the world and what kind of product you're making, you can put those back or are required to put them back. Key targeted examples would be B12 would be one that we would need to take a look at in terms of availability of B12.
Thank you. The next question is, I believe is also for Shannon. This question is, how much is an extracted protein's functionality impacted by genetic versus extraction methods? And can shortcomings in one area be compensated by the other?
So functionality of an extracted protein is impacted by both. And so it is both a nature and a processing method can affect functionality. There's only, at the end of the day, there's only so much you can do with processing in terms of altering the functionality. So if you can if you can bring new functionality to the table with a specific, say, I'm just going to pick pea because that's out there. So if you have a pea proteins tend to function in a few specific ways. If there were pea proteins that could function in different ways, and it gives you a little bit more leverage in terms of how you can extract those proteins and then possibly alter the functionality of those proteins. But the biggest one is your extraction methodology and your drying techniques, and then you can modify proteins in terms of chemical modifications and things like that. And so it depends on your willingness to chemically modify or use chemically modified ingredients. And so that's kind of where some of the genetics might come in as we potentially move towards an industry where we don't chemically modify ingredients to the same degree. You know, some of that can be taken up with new functionalities from a breeding perspective.
Thank you, Shannon. This next question is for Uri. Uri, when you stack or prioritize your traits, are you also applying economics and using selection indexes to focus on profit throughout the value chain?
Thank you for the question. So we are a genomics company. We want and are trying to collaborate with the experts in the field. So if it's a project that involves a few of the examples that I showed or other projects, we do listen to them, and you know the financial assessment comes from the other partners in the project. We never work alone. We're not a breeding company. So if there's a financial assessment required, and there is, they do it and they incorporate that into the need. If a project doesn't make sense financially, obviously nobody wants to start it.
Thank you. This one is for Tom. In the plant protein database, is there data on the sustainability of a specific source, for example, water use?
No, we don't have this data there. However, all of the plant kingdom is much more efficient compared to animal source, and I will be happy to send some resource that we have on that regarding the efficiency in terms of both water, greenhouse gas emission, and land use, and so forth, regarding the use of plant-based proteins compared to animal-based proteins.
Thank you. Okay, our next question is, while mimic of the alternative protein through techniques or technologies, how priorities will be defined between smell, texture, or flavor over nutritional quality? And this one is for Shannon.
This is, for me, this depends on what, so because we service the food industry, and so for me, this depends on what our client or what the customer of that client is looking for and how they would prioritize it. And so we start with texture, then flavor, and typically flavor and smell kind of come together. Just in terms of, and we don't develop the flavors, actually, we work with several flavor houses. And then right now, there's still a lot of work to be done in all four of these. I wouldn't say it's over nutritional quality, but I think we have nutritional quality. There's some aspects there that need to be overcome, like bioavailability of protein, bioavailability of iron, the entrance of B12, so common nutritional hurdles that we need to get over. So for me, it depends on what the client and what the customer is looking for in terms of how we would prioritize it. So it's, I guess the answer to it, the short answer is it depends.
Okay, thank you. And now we have a question again for Tom. Tom, what are the advantages of crop-based over fermented protein sources?
So crops are basically crops are always they're usually much more efficient in terms of their environmental impact and also in terms of cost. Basically, plants are probably the most efficient way to generate food. You just put them in the ground and food grows almost by itself and on the way it cleans the air. However, fermentation is also interesting by itself because the life cycle of a microorganism when we are trying to develop new strains of organisms, the life cycle is much shorter, and because of that, there is an advantage when you are trying to generate something new. Whereas in plants, as you know, it can take several months, almost half a year for each life cycle. So there are advantages and disadvantages for both, but plants are clearly the most efficient tool out there to generate food.
Thank you. Our next question is for Uri. At one point, before being able to use a massive amount of genomic data, there should also be a massive amount of phenotype, analytical data of seed compounds, etc., before being able to develop and use all these genetic markers for breeding. So should there be? Yeah.
Yes. So short answer is yes. Broader answer is that, as I said, we are looking for collaborations with partners. A lot of times our partners come up with the data, they collect the data, they have the ways to create and collect this data, but they lack the capability for genomic analysis. So this is what we're offering is collaborations in which we analyze the genomic data and associate them with the phenotypic data or the actual traits collected either in the field or in the factory. For example, starch content and, I don't know, stickiness of the texture of the meal created from a certain cultivar and such. So this is something that is definitely required and requires also the collaboration of us and the partners involved in projects.
Okay, we have time just for one last question. This question is for any of our speakers. So here's the question. Some of the factors that have been used to propagate plant-based protein are use of antibiotics and hormones with animals. However, isn't the use of hormones, antibiotics, pesticides, fungicides, herbicides equally widespread in plant agriculture?
I can start by saying that the antibiotics that are used on animals are also used for humans, and because of that, that's something that can have a health hazard for humans. Whereas when we talk about pesticides and things like that, we are not talking about the fact that you will have antibiotics in your food. You will eat antibiotics. The issue is that when you use the antibiotics, it causes bacteria to generate resistance in those sites. And that's the issue that we are referring to. Don't remember the rest of the question, so I'll let others answer.
Thanks. Thank you, Tom. Also, I would like to add that through the breeding process, a lot of the projects we are involved in are natural resistances, isolating and creating genetic markers to introduce natural resistances into crops. For example, a virus that disturbs or a fungal pathogen that disturbs lentils or other legumes. There are natural sources of resistance. We identify those regions in the genome and we help through crosses generate a new cultivar that becomes resistant and therefore does not need any of the pesticides or fungicides. So it's actually reducing the need for pesticides. We do not do the same, this type of process is not done on meat. So there's a definite advantage to working with crop-based protein on these aspects.
Okay, thank you very much. So I'm sorry we couldn't answer all your questions, but please note that we will follow up by email to answer all of them after the webinar. I'd like to again thank our guests, Shannon and Tom, and Uri for their time today. Thank you for sharing with us your presentations. This webinar was recorded and therefore will be available in the next few days. So keep an eye out for our email with a link to the recording, and it will also be displayed on our website as well. If you have any further questions or any interesting research projects you'd like to discuss with some of our experts, please don't hesitate to contact us at nrgene.com. Thank you again for joining us today, and I hope you enjoy the rest of your day.